Good morning, good afternoon, and good evening. I think that should cover it. Welcome. Welcome to the University of California Grad Slam competition, also known as the New Knowledge and Innovation Show. I am Teresa Maldonado, the system-wide Vice President for Research and Innovation. It is a great pleasure to be here today to see these students compete. Today, you will have the opportunity to hear from 10 UC researchers, 10 graduate students doing important, impactful work, 10 people solving problems, advancing understanding, and quite frankly, making our world better. These students are representing each of the 10 UC campuses in today's system-wide competition. Their mission, which all have accepted, is to explain their complex research to the public in interesting and accessible ways. Oh, and to do so in 3 minutes. The student presentations are evaluated by a distinguished 5-member judging panel representing higher education, media, arts, and industry. You will learn about them a little later. However, our distinguished judging panel has a 6th ghost member, and that is you. Those of you watching today will have an opportunity after all of the presentations to vote for your favorites. I want to take a moment to thank our wonderful event sponsors. Thank you to LinkedIn for their continued support of UC Grad Slam. In normal times, they host our championship at their San Francisco site. These, of course, are not normal times. We really appreciate their ongoing support and engagement, especially now. A big thank you to Thermo Fisher Scientific for their support. They have generously sponsored all of today's Grad Slam cash prizes. Did I mention that our students are vying for $14,000 in cash awards? We are thrilled to have Thermo Fisher Scientific sponsorship for these prestigious awards. As Vice President for Research and Innovation, I can tell you that the University of California is in the business of knowledge creation, and the students you will meet today are outstanding examples of that. However, I have to say, as impressive as they will be, they are not unique in being talented change makers. We have another 28,000 academic doctoral students similarly engaged. But today is about these 10 remarkable students. So let's get started. It is my pleasure to introduce President Drake, who will MC UC Grad Slam. Dr. Michael V. Drake was appointed the 21st president of the University of California in August of 2020. He oversees this world-renowned system of 10 campuses, 5 medical centers, 3 UC-affiliated Department of Energy national labs, more than 280,000 students, and 230,000 faculty and staff. Dr. Drake previously served as president of The Ohio State University from 2014 through June 2020. Prior to OSU, he served in several roles at the University of California, so he is no stranger to us. He served as 9 years as chancellor at UC Irvine and 5 years as the system-wide vice president for health affairs. An ophthalmologist by training, Dr. Drake received his AB degree from Stanford University, his MD in residency from UC San Francisco. He did his fellowship training in ophthalmology at UC San Francisco and the Massachusetts Eye and Ear Infirmary. Please join me in welcoming Dr. Michael Drake.
Thank you, Vice President Maldonado, and good morning. I'm so honored to MC this year's system-wide UC Grad Slam. The past year has really tested all of us, and through it all, our Grad Slam participants have demonstrated remarkable grit and resilience. In the midst of a pandemic, you continue to invest your time and energy in your education and your research. The University of California is a hub for the creation of new knowledge. Every day, Our faculty and students are making discoveries and developing solutions for our society's most complex challenges. From digital humanities to agriculture, from COVID-19 to cancer, UC research is changing the world for the better. The 10 graduate students we will hear from today are already champions at explaining their research in engaging, dynamic, and understandable ways. They're well on their way to becoming the public intellectuals and ambassadors for research that we all need. So let's dive in. I'm proud to introduce our first campus champion, Adelaide Bernard. Adelaide is a PhD candidate from Belgium, studying metabolic biology at UC Berkeley. Let's hear from Adelaide.
Did you ever wonder why it's so hard to lose weight? It's like your brain doesn't want you to diet. When your body stores too much fat, there's a special system in your brain that's supposed to tell you, hey, don't eat so much. This system relies on a receptor called MC4R. MC4R is a master regulator of hunger. It tells you when you're full. Unfortunately, in some people, this system doesn't work. In the US, 2 out of 3 people are overweight or obese, which is a major healthcare concern. My goal is to understand how MC4R makes us feel full and why it doesn't work in some people. Out of the billions of neurons in your brain, only a few produce MC4R. These MC4R neurons get information from other neurons that sense how much fat you have to then determine how much you should eat. Textbooks tell you that neurons communicate through synapses. This communication is extremely fast, and it works like this. If I were a neuron, I would get information in form of chemicals through a synapse, transform it into electricity to then send the message to the next neuron in the same way. But we discovered that this isn't how MC4R works. Here's one of the neurons that produces MC4R, shown here in purple dots. What we found is that MC4R works in a very specific part of the cell called the primary cilium. Although most of our cells have this cilium, we don't fully understand how they work in neurons. But we do know that when they don't work well, it leads to obesity. And we found that MC4R has to be in the cilium to control hunger. My hypothesis is that if anything goes wrong shuttling MC4R to the antenna, it would lead to obesity. And I discovered that a protein called MRAP2 has a crucial job in this process. It is the shuttle that brings MC4R to the antenna. To prove this, I used genetic tools in mice. On the left, you can see a neuron from a control mouse with normal MRAP2. And on the right, you can see what happened when I deleted MRAP2. Without MRAP2, MC4R couldn't get in the antenna anymore. These mice were hungry all the time and they became obese. This proves that MRAP2 shuttling MC4R into the antenna is absolutely necessary to control hunger. My findings shed new light on how neurons exchange information and specifically on how primary cilia control hunger. This discovery is super exciting because it's going to allow us to better understand why it's so hard to lose weight and hopefully find new ways to fight obesity.
Thank you, Adelaide. Those are fascinating insights on appetite and the brain. Next up is Eric Johns, a PhD candidate and musicologist at UC Riverside. I'm a lover of music myself, and live music is one of the things I've missed most over the past year. Fortunately, learning from a music expert is the next best thing. Eric?
How does race affect our understanding of music? How does white supremacy affect our understanding of music history? Today, I want to talk about how both race and racism not only affect how we understand music history, but can actually change the sound of music itself. And to do this, I want to talk about tango, most commonly associated with the country of Argentina. In the 1895 Argentine census, it declares the almost complete and total disappearance of indigenous and Afro-descendant people from the country, where they are, quote, forming a new and beautiful white race here in the Americas, end quote. This message of disappearance is so pervasive that when I first arrived in Buenos Aires, I was told, aquí no hay racismo porque no hay negros. Here, there is no racism because there are no Black people. And when I asked a follow-up question, I was told that every single person of color I saw was either a migrant or a tourist like myself. Official Argentine statistics today state the nation is 97% white. That is a number only possible through a century of invisibilizing Afro-Argentine history, culture, and existence. As I began my archival research, I began to see names and faces not included in the books. Tango guitar players were often Afro-descendant. In fact, Carlos Gardel, the most famous tango singer of all time, was regularly accompanied by two Afro-descendant guitarists, Jose Ricardo and Guillermo Barbieri. With Gardel, they were stars. Their names appeared on marquees, on the radio, and in film, until Gardel's death in 1935, when their names began to disappear. Not only were their names erased, but the actual sound of their instruments was erased and replaced with the sounds of an all-white orchestra. So what I'm about to play for you will be an original 1928 recording where you'll hear Ricardo and Barbieri. Then it's going to transition into a re-released recording where the version you would hear today, where they have been replaced by the all-white orchestra.
Bueno, me pusiste a la miseria, me dejaste en la palmera, fui afanarte hasta el color.
By studying music alongside race and racism, we can begin to see just how deeply white supremacy goes. It doesn't just erase names from history books. It goes further than that. It can even erase the sounds of voices and instruments themselves, all to fit a white supremacist sound. So the next time you're listening to a piece of music, ask yourself, what part of this has been erased? What part of this story goes unheard? Thank you.
Thank you very much, Eric. What a fascinating journey into the past and origins of the tango. Appreciate it. Shana Bennett is a PhD candidate studying applied mathematics at UC Merced. Shana is using math to solve real-world problems that communities around the world face every day. Shana?
Each year in the United States, invasive species cause over $120 billion in damage to natural resources. That's almost twice the education budget. Often, invasives arrive as unintentional cargo, like an insect riding on a truck to California. A new invasive species arrives every 6 weeks, and California spends $80 million annually trying to control them. But here's the thing. Most control measures come too late because the time between identifying an invasive species and containing it is long. What if we could predict the spread of an invasive species and contain them faster? I'm working with Dr. Khatri in the Applied Mathematics Department at UC Merced to answer this very question. Mathematicians understand that in a simple landscape, let's say a field, a species spreads in a process called diffusion. Diffusion is what we see when we place dye in a glass of water and let it sit for several hours. We've all seen this, and because we understand the process, we can predict the spread of an invasive species in this landscape using a single equation. But as you and I know, a real landscape isn't just a field. It has rivers, roads, mountains, and a species will spread differently depending on what it encounters. This means instead of one equation, we need many, and they have to work together. This is hard. In my research, I'm developing the mathematical and computational tools to solve these equations and allow us to predict the spread of invasive species in a realistic landscape. Let me show you an example. In the top left, I have a landscape with a mountain range. The location of an invasive species is shown in red. On the right, I've taken a snapshot of where my model predicts the invasive species will be in the future. By knowing where the invasive species starts, I can tell where it'll be in a week, or a month, or a year. This is exciting because it means if you're a farmer, my model can help you protect your crops by telling you which land is most likely at risk. And if you're an environmental agency, my model can help you decide which containment strategy would be the most effective. With this tool, we finally have a chance of controlling invasive species quickly and preserving not only our beautiful environment, but our agriculture. Why am I so happy right now? Because I've developed a powerful tool that will help people in our state and our nation stop the spread of invasive species in its tracks.
Thank you, Shana. I can't wait to see how your model is applied to California and actually beyond. Our next presenter, Tori Klein, received her PhD in chemistry and biochemistry from UC Santa Cruz in 2020. Like many of today's researchers, Tori has found useful new insights in unexpected places. Tori, please.
Here in Santa Cruz, we're no strangers to foraging for mushrooms in the redwoods, hunting for the best chanterelles and porcinis that we can find. While those do sound pretty good, if I were out hunting for mushrooms, I'd much rather find one of these. These are just 3 of the many species of cordyceps fungi found throughout the world. Some of you might recognize this name or any of these images from the Planet Earth special about how the cordyceps fungi takes over the minds and bodies of insects, turning them into zombies. And while this might not be the first thing you'd want to find when you're out hunting for mushrooms, these prized fungi have played a key role in traditional medicines for centuries. These days, you can even go online and order yourself a pack of energizing cordyceps supplements. Now, I can't promise you that these mushrooms will boost your endurance as their package claims, but I can tell you that as a graduate student in chemistry and biochemistry, I study how compounds from these mushrooms can help us cure cancer. Now, thankfully, as an organic chemist, I know how to make these compounds in my lab, so I don't have to spend my day babysitting a horde of angry zombie ants. Ooh. Once I've made these compounds, I use a technique called fluorescent microscopy to investigate how compounds from these mushrooms affect cancer cells. Now, many tumors grow because their cancer cells are making too many proteins too quickly, leading to wildly out of control, deadly cell growth. By using a microscope with a special camera and cells whose proteins glow green, I can measure the amount of proteins that cancer cells are making. From images like these, I've determined that my compound stops the dramatically out of control cell growth by stopping this protein synthesis from happening. I've also discovered that my protein compound does this by blocking the conveyor belt that brings in the needed building blocks to the protein assembly machine. During my PhD, I've made new versions of this compound that are even better at getting into cancer cells, which make them a safer and more efficient way to shrink tumors. Now, if you think that trying to find a cure for cancer in a cordyceps-infected, Thailand living click beetle sounds more like a fictional movie plot than reality, you're not alone. But you would basically be describing the plot of the 1992 movie Medicine Man with Sean Connery. But this idea is far from science fiction. In fact, almost half of the drugs approved by the FDA between 1980 and 2014 came from nature, just like my compound. A breast cancer drug listed as essential by the World Health Organization comes from the bark of this yew tree. A painkiller 200 times stronger than morphine comes from the back of this poisonous dart frog. And a lot of us know that the life-saving antibiotic penicillin comes from the mold that grows on your bread. For this reason, I hope my research not only helps to promote biodiversity, but also treat patients with cancer. Thank you.
Thank you very much, Tori. I'm feeling inspired. Those insects are a little bit scary, but I appreciate what you're doing. Next, I'm glad to introduce Anche Fen. Anche received her PhD in molecular biology from UCLA. Anche?
I am a liver transplant surgeon. It's always my greatest fulfillment when the patient restores their health and get back to the family. But sometimes transplants are rejected and there's nothing we can do. This is because we don't completely understand how our immune system is regulated by our genetic code called DNA. 40 years after the first discovery of DNA, scientists finally decoded a human genome. The code comes out to be 4 fairly simple letters, A, T, C, and G. To build a human, you need 3 billion of them. As the most complicated creature, it's not surprising to have such a huge genome. But what surprises us is that only 1% is informative. So what about the other 99%? If they're not informative, then why do we carry such a huge amount of useless DNA? If we stretch out the entire genome, it's over 6 feet long. This is the social distance we have practiced for the whole year. How could this possibly fit into a single tiny cell? Actually, our DNA is extremely condensed and packaged into a substance called chromatin. The DNA is not squeezed and packed randomly like a bowl of messy spaghetti. Instead, it is folded into a very organized structure, territory by territory. Because our DNA is folded so tightly, not every part can be read. And my research focuses in on how the structure of DNA changes in the immune response. In Batman, when Gotham City is in danger, they turn on the bat signal, and it's showtime. Similarly, our immune cells must turn on the danger signal in the form of changing their chromatin structure to initiate the defense system. Let's do a small exercise. Can you see what's in my hand? Can you see the bat signal in my hand? Yes. So whether the chromatin is open or closed affects the activity of our immune cells. This is what we call the chromatin regulatory system. Amazingly, I have found the DNA regions originally considered useless change their chromatin structure to which could allow our cells to fight off the deadly invaders. By sequencing the whole genome, I identified over 60,000 of these useful DNA regions, which could be the therapeutic target to reshape our immunity. In fact, the 99% of human DNA, which was thought to be useless, carries enormous possibilities. Piece by piece, I firmly believe one day we will complete the treasure map of human genome, which will guide us to perfectly understand our immune system so we can guarantee no rejection. Thank you.
Thank you, Anshe. Appreciate the way you brought together DNA, brown bears, and the bat signal. Thanks. We've now heard from our first 5 campus champions. We're going to take a brief pause to introduce our distinguished panel of Grad Slam judges. With expertise in higher education, science and technology, community development, journalism, and the arts, our judges are innovators and visionaries in their respective fields. I encourage you to learn more about these outstanding leaders in our online event program. Well, thank you once again to all of our judges for lending their time and expertise to this event. Next, we will hear from Leslie Hyde, a PhD candidate in the Mathematical, Computational, and Systems Biology Department at UC Irvine. Take it away, Leslie.
Every once in a while, we unlock a potential so powerful, it reaches out and changes humanity forever. In 1953, James Watson and Francis Crick identified the double helix structure of DNA from X-ray images taken by Rosalind Franklin. And in the 68 years since, manipulation of the genetic code has exploded into countless applications and industries. Genetically modified crops, mail-order DNA genealogy kits, new vaccines developed in less than a year to combat worldwide pandemics. And still, we have only barely begun to understand the staggering complexity of this tiny but immensely powerful molecule. Within each of us are trillions of identical copies of DNA encapsulated in the nuclei of our cells. Liver cells, brain cells, skin cells all have the exact same blueprints necessary to make every protein our bodies could ever need. But if every cell has the same master plan, why do they all act differently? Epigenetics and DNA methylation. As an embryo develops, it reaches a point where the cells begin to specialize, becoming a heart, a nervous system, a skeleton. Methylation is when methyl groups attach at various points along the DNA strand to either prevent or promote the expression of specific genes. For example, muscle cells don't need the branch-like structures of nerve cells, so those genes are suppressed. Once a cell has been methylated, after cell division, the children of that cell are usually methylated the same way. But what if they weren't? There's a narrow window of time where each resulting child cell is only half methylated from the half of the DNA that came from the parent cell. The newly added complementary DNA is unmethylated, but only until new methylase enzymes attach. Now, what if we could stop the process or even redirect it? It may be possible to essentially rewrite a cell's fate to become a different cell. The problem is, no one has been able to identify new cells at this key cell fate juncture. Current technologies cannot pinpoint this narrow window because their DNA libraries come from cells at varying stages in the cell cycle. That's where I come in. Determining the age of cells based on their position in a biological process is known as determining their pseudotime. No one has yet developed pseudotime based on methylation signatures. My research is to quantify methylation pseudotime in order to identify newly formed DNA strands. Once identified, a whole host of new targeted experiments can be performed with the ultimate goal of redirecting cells to perform different tasks. Someday, from a simple swab inside a patient's cheek, we could grow a replacement liver or kidney, or replenish bone marrow with a perfect match. My work will be a vital step in unlocking this potential. And once we succeed, humanity will be changed forever. Thank you.
Well, thank you, Leslie. What a fascinating journey. Zat. Our next campus champion is Haleh Yazdi. Haleh, is a PhD candidate in psychology at UC San Diego. Her research will take us around the world for a new perspective on childhood.
When I was 3 years old, my family moved to a rural city in the South called Commerce, Texas. And it didn't take long for me to realize that my Iranian family didn't exactly blend in with the other families there. This is because early in life, children recognize similarities and differences between themselves and others on a variety of traits, like gender and skin tone. And they use this information to categorize people into preferred in-groups and disliked out-groups. This process over time can lead to discrimination against out-group members, which is something that my family experienced. Now, a popular theory in psychology is that children operate on the belief that what's similar to me is good and what's different from me is bad. But almost all the research in this area comes from Western societies, leaving it unknown whether children across the globe develop prejudices in this way. My research takes a more global perspective towards studying childhood prejudice. Take Iran, for instance. Iranian children know that they're very different from Americans and that their country has ongoing tensions with the US. But they also know that the US has high status globally. So how might these children weigh similarity against other factors when deciding who they like? To answer this question, I asked children in Iran how much they wanted to be friends with members of 4 different groups. Iranian children from their own school, so their in-group, Iranian children from a different school, so an out-group that differed on one dimension, Arab children, a group who is very similar to them with regards to language, religion, and proximity, but one that they've historically had conflict with, and American children, a group who they're very different from, have current political conflict with, but a group that has high status globally. What I found was that Iranian children expressed the most desire to be friends with American children. In fact, so much so that they rated them more favorably than their own group. Surprisingly, children also expressed the desire to trust and be loyal to Americans. On the other hand, Iranian children expressed the least desire to be friends with Arab children, a group who they perceived as having low status. My findings show that children will in fact welcome friendship with a group who is very different from their own. But it's our responsibility as adults to encourage these friendships and to first teach children that all groups have equal status. Once we do this, we can eliminate prejudice early in life and promote better, more peaceful relations in our country and across borders. My research brings us one step closer to a world where we see one another as equals.
Thank you, Hele. It's clear that this research has really important implications for combating prejudice really around the world. Thank you very much. Next up is Anthony Bonita. Anthony is a PhD candidate in the Biomedical Sciences program at UC San Francisco. Like many research scientists at UCSF, Anthony is exploring ideas in areas with important implications for patient care. Anthony?
A show of virtual hands. Who here knows or has lost a loved one to cancer? Same. It is indiscriminate and will claim millions of lives this year. In the past decade, significant progress in cancer therapy has prolonged survivorship and has saved lives. And a big contribution to this success is immunotherapy, in which scientists have manipulated the power of your immune system to prevent, control, and eliminate cancer like these ninja cells. One way that this can occur is through the recognition of danger signals. They become localized to the surface of the cell, which the immune system can recognize and eliminate. But one type of cancer that does not respond to this type of therapy is pancreatic cancer, one of the world's most lethal diseases. So my mission became about understanding how these tumor cells have become so adept at surviving despite all the checks and balance systems that our body uses to keep us from getting sick. So I read a lot about this cancer and found something unique. There are high levels of activities from the structure called the lysosome, which are these little digestion machines that eat material similar to Pac-Man. So how do you study what a pancreatic cancer cell is eating? So there must be a reason why these tumor cells have so many lysosomes. So I set out to study what makes them so special. But how do you study what they eat? You design a tag that specifically recognizes only those lysosomes, isolate them from a cancer and a normal cell, and compare the contents of what they ate. I did just that and found some pretty striking results. These pancreatic cancer cells were selectively targeting certain proteins for digestion. In other words, they became picky eaters. And get this, when I was screening through the list of proteins that were selectively targeted to that cancer lysosome, guess what I found? That danger signal. It was no longer eaten up. That danger signal was no longer on the surface of the cell, but rather getting eaten up by the lysosomes, allowing them to hide from the immune system. So I blocked activity of the lysosome and found that the digestion machines no longer ate that danger signal. They were instead rerouted to the cell surface, allowing the immune system to once again recognize them. Remember I mentioned earlier on that pancreatic cancer does not respond to immunotherapy? After implanting pancreatic cancer cells into mice, immunotherapy alone had no effect. But when used in combination with blocking activity of the lysosome, the mice lived much longer than the untreated ones. The most exciting part is that I found a potential answer to significantly extend the lives of patients, where over 70% live less than a year after diagnosis. My hope is that this will be translated to a clinical trial in the near future that will benefit not only patients that suffer from pancreatic cancer, but all other types of cancers that have become picky eaters.
Well, thanks very much, Anthony. Fascinating work, and wish you the very best. Encouraging to hear about the future of pancreatic cancer treatments. Our next presenter is Amelia Munson. Amelia is an animal behavior PhD candidate at UC Davis. Amelia?
All of my favorite stories about me from my childhood involve some amount of danger. They're the times when I fell out of a treehouse and then got right back up again. And I think I like these stories so much because these are the moments that made me into the resilient adult that I am today. And I think this is a pretty common narrative. But interestingly, it's not the logic that we use when we are tasked with taking care of some of nature's most valuable babies, endangered wild animals. Take salmon, for instance. When we raise these culturally and economically important fish in captivity, we raise them in very sheltered hatcheries. And then we release them into the wild where they have to deal with real predators that really kill them. I was interested in whether there were developmental experiences that we could give fish that would improve their ability to deal with challenges. To test this, I exposed baby Western mosquito fish to different developmental stresses, basically the fish equivalents of falling out of a treehouse. So I have this red group that I exposed to changing temperatures, this blue group that I exposed to the very scary scent of largemouth bass, the purple group that got both, and the golden group down at the bottom that wasn't exposed to anything stressful while they were growing up. And then I looked at what effect these experiences had on the fish as adults. And some of the effects are obvious just by looking at them. You can see that my middle two groups, the ones exposed to predators, grew up to be smaller, which is interesting because smaller fish, less appetizing to predators. But I actually cared less about what my fish looked like and more about who they were. Basically, I wanted to give them a BuzzFeed personality quiz, but tailored towards fish. So I took all of my fish and I put them into new safe tanks and looked at their behavior. Interestingly, my group that got both stressors during development, they were more active during this pulse of safety, suggesting that they could gather resources to use for future challenges. But the most interesting thing I found is that exposure to any stressor during development resulted in fish that were more consistent. And this is fascinating because consistency is a general pattern of behavior that helps fish coordinate group behavior and do other anti-predator defenses. It seems like developmental stress is crucial to making a fish behave like a fish. My fish are a great reminder that what doesn't kill you doesn't just make you stronger, it makes you normal. Thank you so much for listening.
Well, thank you, Amelia. I feel that this is important research with important lessons for not only fish, but all of us. I know some kids who are raised like your goal group. No comment. Last but not least, I'm proud to introduce Logan Kozel. Logan is a PhD candidate in the Department of Ecology, Evolution, and Marine Biology at UC Santa Barbara. Logan?
Our oceans sustain us, and every year the world relies more on marine farming or aquaculture to provide protein to its growing population. However, marine species are under acute threat from heat waves which are increasing in both frequency and duration. One such species is the green shell mussel, a species of immense cultural and economic significance to the country of New Zealand. Green shell mussels make up over 85% of New Zealand's annual aquaculture export, but recent heat waves have literally cooked thousands of mussels in their shells on New Zealand's northern shores. So my research investigates whether parental experience can mitigate the effects of marine heat waves in aquaculture across generations. If an adult animal, such as a mussel, experiences an environmental stressor, particularly while forming their sperm or their eggs, this can induce a change in performance in their offspring when those offspring experience the same stressor in the future. These transgenerational effects can be driven by maternal provisions, things like lipids and proteins, which can pass directly from mom to offspring through her eggs. Epigenetic modifications can also transfer from parents to offspring and influence future gene expression. While epigenetic modifications do not change the DNA sequence itself, they can change the way that the DNA is packaged and regulated, influencing which genes are easily accessible for enzymes to transcribe and express. So I want to show you one way that this can play out. I conducted an experiment where I exposed female mussels to either control or marine heat wave temperatures during the summer, and then allowed some of the heat stress mussels to recover prior to reproducing, while some remained at high temperatures. Then I assessed the development of their offspring to see how they coped with the heat stress themselves. My results showed that the offspring, which came from mothers who experienced a marine heat wave event, but were allowed to recover prior to reproducing, had significantly higher normal development than those from control or heat-stressed mothers. This means that farmers can actually improve the capacity of their mussels to endure future heat stress by giving the parents enough time to recover after a heat wave event before breeding them in hatchery. Aquaculture provides a really unique opportunity to manipulate the parental experience and to actually treat the two sexes differently in ways that are not possible in wild populations. By studying the physiology of green shell mussels and other species, we can leverage their own biology to help them, and in turn us, be more resilient in the face of climate change.
Thank you, Logan. It's inspiring to hear about innovative strategies for protecting this important wildlife population and its environment. And really, to all 10 Grad Slam champions, I hope you're proud of your remarkable achievements and your contributions in these broad fields. Your research is really a reflection of the new knowledge being created at the university every day. So thank you and congratulations really to all of you. Terrific work. It's now time for one of the most important parts of today's event, voting for the People's Choice Award. Ta-da! Our audience is going to have the chance to vote for your favorite presentation during today's Grad Slam event, and the person who receives the most votes will receive the coveted People's Choice Award. So to submit your vote, you visit ucal.us/gsvote, or you can vote directly from the UC Grad Slam homepage. Voting's now open and will only be open for 5 minutes, so don't delay, vote now. Once all the votes are counted, the winner of the People's Choice Award will be announced live, along with the other awardees, including the winner of this year's system-wide Grad Slam. So stay tuned. And while the voting's taking place, I'm excited for an opportunity to talk for a bit more with today's competitors. And this will be great, and have a nice chance to say hello. And again, let me say congratulations to all of you. I can't wait to learn even more about your research. And maybe let's begin with Adelaide. So Adelaide, what got you interested in studying biology, and where did your passion come from?
Hi, thank you for that question. You know, as a kid, I would always ask a lot of questions about my surroundings, and my parents were always sad, telling me, oh, I wish we were doctors and we could answer your questions. Of course, we didn't have so much internet like we do now. And, you know, I had a great biology teacher growing up, and we were a very small class. So it was kind of private lessons, I guess. And it was just fascinating to learn about physiology and the human body. So I decided to go on and study that in college. And, you know, I just loved specializing in physiology and then more of a metabolic and neuroscience. And I'm really happy that I found the subject that I'm studying right now for my PhD thesis. It's just the perfect match for me.
Well, it's fantastic and great to see where that early passion has taken you to. And maybe same question, Erica, how about you? Have you always been a music lover? And I'm interested in whether or not the music took you to the social and political circumstances of the people you were studying, or the other way around.
Thank you for that question. Actually, weirdly, no, I wasn't always a music lover. I grew up in a house filled with music, but for some reason, I really hated it when I was a kid. That thankfully and obviously changed. And yeah, I grew up in South Louisiana, a place that has incredible music, but also incredibly messed up politics. So I started seeing these things while I was beginning my research in tango, and it just kind of, I went down the rabbit hole and it revealed itself to me. I didn't go looking for it. It became very obvious to me by studying it.
You know, it's fascinating the way that art reflects people and what they're wishing to express to us, and again, the social and political and personal circumstances of the artist. And it's great to make those connections in all forms of art. So I appreciate that.
Absolutely.
Let me go on to Shana. Tell us a bit more, Shana, about how you came to study mathematics and what's meaningful about mathematics to you.
Yeah, of course. Thank you. I originally started out in environmental science as an undergraduate. And during that time, my undergraduate advisor was recommending to all his students that we take more classes in mathematics. And I was homeschooled K through 12. And during that time, I really didn't understand mathematics after Algebra 2. And I was lucky enough during my undergraduate studies to have some really great professors. And I just started to find the field fascinating. And during my senior year, when I had to start thinking about what to do next, I was really interested in how can I take my background in environmental science and my interest in math and combine it. And I was lucky enough to do a research experience for undergraduates at UC Merced with Dr. Khatri, who's now my PhD advisor. And here I am.
That's fascinating. And your story reminds us so much of how important early mentors and guides and stimulators and supporters are. So great to hear that. Tori, I'd love to hear more about your journey into the sciences and how you came to study zombie insects.
Yeah, thank you. It's an interesting story. I grew up really wanting to be a dolphin trainer for a long time. And then, so I always really loved nature and the outdoors. And then I actually watched Medicine Man, the Sean Connery movie, with my dad a long time ago when I was a little kid. And I was like, wow, that's really cool. I wonder if this is just a movie. And I forgot about it for a while. And then eventually I had a class in my undergrad where we learned about marine natural products and all about these different amazing medicines that you can get from nature, some that are used now. And I just thought, wow, this is a really cool way to combine the two things I love of marine biology and wanting to grow up and be like Sean Connery and go out in nature and find cures for cancer. So, really was apparently inspired from a movie that no one else but I have seen. So
I hate to spoil, but I saw it too. And I thought it was great. And I think it's fascinating that you wanted to grow up to be like the scientist Sean Connery rather than the spy. So I think that's a good thing. Good to see you.
Thank you.
Anchen, how about you? How did you develop and hone your interest in biology?
Yeah, actually, when I was a kid, I just wanted to become a doctor, to be honest, initially. But my family elderly just encouraged me saying, oh, if you become a surgeon, you just save one life at a time. So during your whole life, you probably can save 1,000, but not exceeding more than like 1,000 or 2,000. That's the maximum amount. But if you do research, then you can save more. So that's the reason why I'm doing research right now. And I'm pretty sure that I can make only a very small contribution to that. Our future goal of saving thousands of millions of people. But with all these brilliant minds together, I believe that one day we can achieve it.
Well, I appreciate that. And your parents set a pretty high bar for you, but I think that's a terrific work. And, as a medical doctor myself, it's great to be able to combine the clinical and the research and do a little bit of both. So it's terrific work. So maybe a little different question for our remaining presenters. Let me ask this. What trend or development in your field makes you the most excited for the future? Let's start with you, Zat. Leslie, what do you see as the future in your field?
Zat. So the future of my field, I'm working on methylation, and the future of that is looking at the individual differences in our cells and how they generate the individual differences in humanity and also many other creatures. We study murine embryonic stem cells, which are mice, humans, all of the above, and the ability to change and adapt and streamline what we want out of cells and be able to create new organs. And I'm just really excited about everything. It's fun research. I'm happy to do it.
Wonderful to hear. And I want to assure the other contestants that even though I'm still do my Zot, I'm not voting today. So you don't have to worry about that. Appreciate that very much. Halle, how about you? What trends or developments in psychology do you find most promising and exciting?
One of the things I'm most excited about is this new attention to doing cross-cultural research. So previously in psychology, as I mentioned in my talk, there was a focus on Western populations and then generalizing human behavior based on a very limited population. So I'm really excited about this movement towards including other cultural groups and doing more cross-cultural research. It's something I'm really passionate about, and I'm glad that this is now becoming a trend in psychology.
That's very fascinating. Your work is interesting as well, and how people decide who they want to be and where they want to go by looking at the world broadly is a fascinating area of thought, and I appreciate that work. How about Anthony? What are you intrigued by as you think about the future of cancer treatments? You mentioned when you were saying hello, how many families are affected by cancer, and it's really pretty much every family at one time or another. So, and I'm curious about whether or not the global pandemic has changed your perspective at all.
Yeah, I think it's really incredibly important to think about like the current therapies that we have right now. Obviously, with the current global pandemic, we have the vast production of the vaccines, but on the side of cancer development, one of the things that I'm most excited about is cancer immunotherapy, which is one of the things that I did talk about in my presentation, and really understanding why it doesn't work in some cancers versus why it works in other cancers. So that's one of the reasons why I really want to study pancreatic cancer and see whether or not or why pancreatic cancer does not respond to immunotherapy, and what can you do in combination with other FDA approved drugs that have already been approved and that have already been used by companies and patients in the hospital, how you can combine those two kinds of therapies in order to improve the outcome of a cancer where the survival rate of the 5-year survival rate is less than 10%. So hopefully the research that I've done can inform that and help save lives.
Yeah, I think that immunotherapy really is very promising for the future, the way that biological systems normally defend themselves against these insults. And if we can learn how to strengthen and manipulate that, that would be an important way to try to do what we can to really decrease the cancer burden for the world. So good luck with that. Appreciate it. And Amelia, how about you? Are there developments in your field that you feel particularly optimistic about looking forward?
Yeah, I mean, I think a lot of the developments come from this sort of interface between the fact that we are working with animals who are currently living in an environment that is changing really rapidly. So lots of people who study behavior are looking at what the environmental impacts are and how animals are behaviorally shifting and how their behavior is affecting the way that they respond to those changes. So I'm really excited about that moving forward. And I'm actually about to start my postdoc looking at how fish are better able to prepare for changing water temperatures. So yeah, it's a pretty exciting time to be studying animals in the environment.
It really is. And I look forward very much to seeing how this pans out. So thanks very much for what you're doing. And Logan, how about you? What are you excited about in your field as you look toward the future?
I don't know if this is super specific to ecology, but one thing I'm really excited about in my field that I think applies to a lot of other people's as well is that we're at this cool period where we're simultaneously developing so much technology that looking at the mechanisms behind physiological phenomenon are more plausible, you know, like a lot of the awesome things that Leslie talked about. But at the same time, people have this openness, it seems, to think interdisciplinarily and to want to collaborate across like biologists with people who are in aquaculture or in clean energy, and having that ability to look outwards and want to make connections at the same time that other people are drilling down really deeply into the mechanisms, I feel like is a major push for the actual applications that we're going to be able to get out of traditional ecological and physiological research.
Well, fascinating. Thank you. Really, thank you all. It's really been great to hear from you all. And congratulations again on the quality of your research and these really outstanding presentations. Actually, it makes me miss more that we can't be together. I really would love to talk more about these things. And we all, I think, are looking forward to seeing where your research takes you. After hearing from today's presenters, you all can see the difference that UC graduate students are making through their research. And I'm really proud to be able to share a video featuring even more groundbreaking UC research that's making an impact here in California and around the world.
Okay, here we go.
I mean, that's incredible.
We really have to talk about the whole question of time. So the first part is just like...
And it was from here that the first message was sent.
3, 2, 1, and liftoff. Liftoff of Discovery.
We're curious people. We want to know why things happen the way they do.
This is going to be an amazing technology.
Remarkable.
Use mass spectrometry, which allows you
to have the maximum number of input segments.
Declared the COVID-19 outbreak a pandemic.
every day we save lives.
But there's more. What else can we imagine?
Wow. It's always inspiring to hear how UC research is solving, excuse me, some of the world's most urgent challenges. As you've seen today, UC students and faculty are deeply committed to using their knowledge and their research to make the world a better place. All of you give me hope for the future. And now, I will hand it over to UC Provost Michael Brown to announce all of our 2021 Grad Slam winners. Thank you all. I can't wait to see what you do next. Over to you, Michael.
Thank you, President Drake. And welcome, by the way, to the first UC Grad Slam of your presidency.
Thank you.
I want to add my thanks to all of the student competitors for their outstanding presentations. And I can say the world of scholarship is in great hands. Now is the portion of the program where I suddenly become very popular and everyone wants to hear from me because I am here to announce the winners of this year's UC Grad Slam competition. But I got to tease, before we announce the winners, I'd like to take a moment and acknowledge the graduate deans from each of the campuses. These are the individuals responsible for leadership on behalf of graduate education throughout UC. They made this day possible as each of them coordinated successful campus competitions to deliver today's outstanding competitors. They not only host their campus event, but they commit to doing this annual system-wide tournament as well. And so they are Lisa Garcia Bedoya at UC Berkeley, Jean-Pierre Delplanque at UC Davis, Jillian Hayes at UC Irvine, Susan Edner at UCLA, Chris Kello at UC Merced, Sean Bowler at UC Riverside, James Soto Anthony at UC San Diego, Liz Silva at UC San Francisco, Lila Roop at UC Santa Barbara, and Quentin Williams at UC Santa Cruz. Now that we have taken a moment to acknowledge our graduate deans, let's get on to the awards. I want to again thank our award sponsor, Thermo Fisher Scientific, for providing all of the cash awards today. We will announce the People's Choice Award, a third place award, a second place award, and the grand prize winner who will receive the Slammy. I hope you could see this. You have heard of the Grammy, well, this is the Slammy, and it is very coveted. So all of the presenters were extremely impressive, so we know this was difficult work for the judges. Let's hear from our audience judges first, and our thanks to everyone who took the time to cast an online vote. We can now announce the system-wide People's Choice Award, and the winner will receive 1,000 smackaroos. Now the award goes to, and I have it fresh here, Logan Cazal at UC Santa Barbara. Congratulations. Third place and 2,000 smackers goes to Adelaide Bernard from UC Berkeley. Congratulations. The second place and $4,000 goes to Anche Fenn from UCLA. Wow, this is exciting. Congratulations. And it's time to name our winner. This is such a dramatic moment. If this were a reality TV show, we would have to cut to commercial right now. But that's not us. So there is only one thing left for me to do. It is my pleasure to announce the UC System-Wide Grand Slam champion. $7,000, and this coveted Slammy goes to Shayna Bennett. Congratulations. Congratulations, Shayna. UC Grand Slam is an annual event, so we also have a perpetual plaque to award to Dean Chris Kello. Wait a minute. I hope I have this information right. Dean Chris Kello from UC Merced. Do I have this information right? Okay. But really, every one of our deans has plenty to brag about. All of these students were outstanding. I absolutely meant what I said. With researchers like these at the helm, the world and the research that will benefit it is in excellent hands, and I am excited for the future. Congratulations to all of our students who presented at today's system-wide competition, and to our graduate students throughout the system who participated in and supported their Grad Slam efforts. Well done, and thank you. And on behalf of President Drake and all of us in the Office of the President, we want to thank our wonderful partners, LinkedIn, who has been an historic partner with us, and Thermo Fisher Scientific. We couldn't ask for better partners. We really appreciate the work done by so many on behalf of today's event. It took a lot of creativity, time, and talent, and behind the scenes work. Believe me, I just had snippets of this to make this event happen virtually, and we are so happy it has come across so well. There are far too many people to mention by name, but please permit me to permit a shout out to the entire UCOP Grad Slam team. So many of them are in the UCOP Grad Studies department. They are the dream team. They're very special. And I want to also thank the marketing and communications team too. Yes, you all rock. Yes, you do. And finally, I want to thank each of you for joining us for this very special event. We appreciate your wonderful support and the great energy on behalf of graduate education at the University of California. We have given you just a glimpse of what research at UC is achieving. We hope that you will take the energy and the insight from today and help find ways to actively support the graduate research enterprise at the University of California. Funding and engagement are always helpful. And we want to just thank you again very much. And we look forward to seeing you next year